We will now begin the LigaChemBio Global R&D Day 2025. I am Jung Dae-young, head of the IRBD team, and I'll be your host for today. Before we officially begin, we kindly all our participants on site to make sure that your mobile phones are set to silent or vibrate mode. Today's event consists of three main parts. Part 1, titled Current and Future Development Roadmap, will cover the current status of our clinical development programs and our future plans. Part 2, Next Drivers in ADC and Immuno-Oncology, will introduce our new ADC pipelines and updates on next-generation immuno-oncology drug development of LigaChem. Finally, Part 3 will present our global business strategy and corporate outlook. Let me now introduce our executive leadership present today. As your name is called, please stand up and give a brief greeting. Mr. Yong-ju Kim, CEO of LigaChemBio. COO and CFO LigaChemBio, Park Se-jin. Mr. Yong-nak Cho, Senior Executive Vice President, Head of Development. Senior Executive Vice President and Head of Global Business Development and President of ACB, Choi Jae-uk. Dr. Jung-chul-uk, Head of Discovery Research. Mr. Seung-dok Chang, Executive Director and Head of Management Support Center. LigaChem's Executive Director of New Business at Orion and Internal Board Member of LigaChem, Mr. Hyung-seok Kim. Mr. Seung-ho Lee, CEO of Daily Partners and External Board Director of LigaChemBio. Last but not least, let me introduce our key research leaders. Dr. Chang-sik Park, Head of the ADC Research Center. Dr. Suk-young Jang, Head of the Immuno-Oncology Center. The leaflet you received includes QR codes for downloading presentation materials and submitting questions. Presentation materials will be available for download after the morning and afternoon sessions. If you have any questions during the presentations, please submit them via the QR code and we will address them during the Q&A sessions. All the morning session will feature English presentations only. We have translation receivers placed in front of you. Please utilize them. We will now begin the event with opening remarks by our CEO, Mr. Yong-ju Kim. Good morning, ladies and gentlemen. This is our second R&D Day event. Thank you very much for the great turnout, and also I appreciate your great interest in this event. We were supposed to begin earlier, but we had a lack of communications with you, so I apologize for that. Today, as was introduced by our MC and host, so most of our discussions will be about our R&D except for our business plans. So briefly, let me summarize what we are going to cover. As you see here, except for the business plans, everything else will belong to one of these three categories. One of the most important part is that we are going to have as many pipelines as possible. So we are making very aggressive approach. There are 5 pipelines in clinical trials, a total of 27 will be under development, and about 15 will be IND stages. So we are going to actively pursue clinical trials. So we are making a multifaceted approach, and also we are going to develop business development. Not only ADCs, but also modulate payload-based ADCs. So we've been preparing for these for years already. At the World EC Asia Summit, there were 3 keywords. One is the ADC, a bispecific ADC. Second, and bio ADC. There was a third one, PDC, peptide conjugate. So peptide conjugate is emerging again. Many people have negative thoughts about peptide, but peptide has actually a lot of advantages. So I'm a bit hesitant to talk about this. We are actually preparing for this. For the next 3 years, we are going to focus on these 3 categories. Aside from that, we are also focusing on novel targets, in addition to the clinically proven targets. So target development speed will be a lot slower down in the future. So I believe first-in-class novel targets will be identified soon. Second, regarding platforms, we continuously improve our platforms. Why? First, although we say customized linker, depending on the payload, properties and characteristics are different. So depending on the payload, we need to customize linkers. So when we have a new target, we are going to have a payload TOF. So although there's a mainstream movement, or statin, PBD, DNA-related stuff. So there are many different types. And also, we need completely new payloads. So if you continue to use one particular payload, it will develop resistance. So you need to change a payload. Since we are dealing with many different payloads, we need to develop the linkers, relevant linkers. So we have to continuously improve them. Second, I call it NOTA. This is very important. So far, there have been high DAR. So as you attach more payloads to antibody, the original properties of antibody will be lost. So PK will be worsened. So if you double DAR, that doesn't mean that efficacy will increase twice. It might rather increase toxicity twice. So low DAR, if you do that, TI will be further widened. So we are going to utilize low DAR. Of course, we can attach many drugs to antibody, but we are trying to pursue low DAR. And committed therapy, that's very important. ADC and IOC, there have been many clinical trials. So there should be a scientific rationale. So because of that, we are developing many different ADCs and pipelines. So we are studying many different studies for new mode of actions, for combination therapies, new payloads. Since the founding of our company, this is our ongoing task. There are only 3 payloads clinically, so we need to identify a new payload, although it's arduous and lengthy process, but it's a must. Because if you continue to use the same type of payload, it will develop resistance. So not only STING. So we have all the backgrounds related to IO, and we are developing small molecules related to IO. So together with our partners, we are working together. Sometimes we already developed enough new mode of vaccine, new payload. We are identifying them and developing them. We will continue to do so. Regarding TPD, there's a limitations of TPD, especially for hematologic cancer. It can be utilized a lot. So from many different angles, we need to develop TPP, and we are working hard to develop TPPs from many different perspectives. So I believe our speakers will discuss that, especially in the later part of our event. So we are developing, we are growing, we are running our business. And regarding our business development plans, Mr. Che will give you details about it. Thank you very much.
Let us move on to the first presentation. As part of the current and future development roadmap, our first presenter is Robert Lutz under the topic of the ADC explosion, the expanding landscape of ADCs. Let me introduce Dr. Lutz prior to his presentation.
러츠 박사는 당사의 임상개발 부문 최고
Dr. Lutz is
자문위원이자
our Chief Clinical Development Advisor and currently serves as the CSO of our partner,
이전에
Exuda
현재는
Therapeutics.
에보비사로 인수된
He is
이뮤노젠에서
a
23년간
leading
재직하며
authority in the
난소암
ADC
치료제인
field,
엘라히어,
having spent
유방암
23
치료제인
years
테사엘라 등
at
FDA
Immunogen,
승인 ADC 치료제 개발을 주도하신
now
ADC
acquired
분야의
by
최고
AB, where
전문가이십니다.
he led the
러츠
development
박사는
of
오전 세 번째 발표를
FDA-approved
진행하실
ADC
예정이어서
such as
Q&A는
Elara for
러츠
ovarian
박사의
cancer
세
and
번째
Casella for
발표가
breast
끝난 후
cancer.
한 번에
He's
진행하도록
also
하겠습니다.
going to give us third presentation,
Dr.
so
Lutz,
there will be Q&A session at the end of the part
I think
one.
that Mr. Kim already finished his time, so you got extra time. So,
Oh, great.
yeah, please start your presentation.
I will do. I'll look to share my slide right now.
Not yet.
It takes 30 minutes to
It's
go
okay
from
now.
Boston to Seoul, I think. Yes. Do you see my title slide?
You can see clearly.
Oh, great. Well, hi everyone. It's my great pleasure to speak to you all today, and I apologize for not being able to present to you in person. In this presentation, I would like to provide an overview of the expanding landscape. Just one second, technical problem here. There we go. I would like to provide an overview of the expanding landscape of the ADC field and introduce some of the opportunities and challenges that we face in thinking about the development of new ADC therapies. I characterize the past 5 to 6 years in the field as an ADC explosion, as noted in my title. In large part, this is due to the success of Enhertu, a HER2 targeting ADC for the treatment of breast cancer and other HER2-positive cancers. While interest in ADCs has existed for over 30 years, and I'll note I've been working on them almost that long, it was Enhertu that redefined what scientists considered could be possible for ADCs and has led to an explosion in effort and interest in the field. One way to look at the explosion is to map the number of clinical trials started per year. And starting as early as 2000, the number hovered between 10 to 15 clinical starts per year. A slight inflection came with the first ADC approval for Adcetris, and as clinical data emerged from clinical trials for TDM1 or Kadcyla, again, some increased of interest. But it was emerging clinical data from Enhertu that really put the effort on the modality into overdrive. Additional approvals have continued to feed that momentum. So the profile is similar if you look at the number of ADCs entering their first clinical trials, with over 62 ADCs starting clinical trials in 2024. This includes a surge of ADCs with topo-1 inhibitor payloads following the success of Enhertu, which is comprised of the topo inhibitor payload deruxtecan. In fact, most ADCs in the over 1,600 clinical trials currently ongoing use either a tubulin or topo-1 inhibitor as their payload. And this includes about 80 ADCs in phase 3 clinical trials. This large number of ADCs in late-stage trials predicts a continued substantial number of ADCs over the next several years. The excitement in the ADC space has not gone unnoticed by private and public funding interests, as well as interests in licensing and M&A opportunities. The number of ADC deals, either technology or asset licensing, or M&A deals, have averaged about 100 per year over the last 4 years, with total deal values reaching between 20 and 150 billion US dollars. And the global footprint of companies involved in the development of ADCs continues to grow, with over 1,200 biopharma companies currently involved in ADC R&D across all sizes, from small biotech to large pharma, with the top three locations for ADC R&D companies being the US, China, and Korea. So what's the big deal about the ADC space? Why is the commitment to the modality worth it? What is unique about ADCs that support continued investment in the space? So first of all, I think it's important to recognize that cancer still has an enormous unmet medical need. And even with all the advances in cancer therapies, most patients with cancer still die of their disease and spend the rest of their lives dealing with their disease and the side effects of their efforts to fight the disease. But still, why ADCs? I think the simple answer to that question is that ADCs are one of the few modalities that rely less on the biological properties of their targets and more on the characteristics of their design. This is drastically different from other modalities such as immune checkpoint modulators or other unconjugated antibody therapeutics. For this latter group, success is largely dependent on the biological hypothesis being correct. Is this target good for immune checkpoint inhibition or the like? If that hypothesis if that hypothesis is correct, and if I block the function of this protein, something good will happen. But if the hypothesis is not correct, there is very little learned to help with the next attempt. All that meant was my hypothesis about the biological nature of that target was not appropriate. And so it's back to the beginning. For ADCs, however, the designability of the modality means that prior experience increases the probability of success for next generation compounds. We can understand what the limitations of the current generation compounds are and innovate to find improvements that improve outcome. So, in other words, the strength of ADCs is that they are designable drugs. And indeed, while they're complex molecules, this complexity offers vast opportunities for optimization and differentiation. So new concepts, technologies, and designs are emerging all the time to address the shortcomings of prior design and exploit the learnings of the preceding generation of compounds. And in fact, the field is addressing all aspects of ADC design. This may include discovery of novel targets, for example, novel epitopes, or understanding the properties of ADC targets to manipulate them in a different way. It may include novel innovations for the binders, the antibodies, the alternative types of binders. Also, improvements in bioconjugation technologies for ease of development, improved biophysical properties, and improved stability. Novel linker chemistries are being developed to influence and improve the properties of the ADCs, for example, impacting its interactions or impacting the way in which it's activated, that we'll hear about more tonight. And it also may include improvements in payloads to overcome resistance, improve tolerability, and in particular, of interest to me, explore new mechanisms of action. And the importance of ADC design is no longer a hypothesis or an assertion. The impact of ADC design has been clearly validated in the clinic. And many of you who have been following the field will recognize this example. A simple comparison of TDM1 and TDXD highlights the importance of ADC design. TDM1 was first approved in 2013, with currently about $2 billion in annual sales. TDXD followed with first approval in 2019, 6 years later, and sales are expected to end up well over $10 billion a year. Both are directed against the same target, HER2, and utilize the same antibody, trastuzumab. So beyond that, the designs are very different. And it's clearly in those differences in designs that one sees the difference in these compounds. For example, TDM1 uses lysine conjugation chemistries. TDXD is conjugated to cysteines using classic maleimide chemistry. TDM1 has a non-cleavable linker, while TDXD has a peptidase cleavable linker. One, TDXD shows bystander activity, which may improve the activity in heterogeneous tumors, while TDM1 has little bystander activity, but that may provide improved tolerability compared to other designs. And potentially the most important difference for these two ADCs is that TDM1 has a tubulin inhibitor payload, with TDXD delivering a topoisomerase I inhibitor payload. The impact of the differences in the ADC design are profoundly clear when comparing the anti-tumor activity, with TDXD having a much higher response rate and progression-free survival in a head-to-head clinical study. They're not really very close, as it turned out. This level of response for TDXD at the time was unprecedented, particularly for solid tumors. But it is important to note that this improved clinical activity did come at the cost of significant hematologic, lung, and GI toxicity. And this latter fact highlights the need for continued innovation in the ADC field. So, what about other aspects of ADC development? And let's take a look first at the landscape of ADC targets that are being investigated. It's quite impressive, actually, the number of targets that have been evaluated over the years for ADCs. Over 100 targets are currently being explored clinically by at least one clinical ADC, with over 200 targets having been explored across cross time by at least one ADC. Now, some of the ADCs to these targets have been discontinued, and many discontinued targets have no current disclosed efforts. But interestingly, many targets with successful clinically validated ADC programs have had discontinued programs in the past as well. This again highlights the importance of design, innovations, and learnings from prior generation molecules. So just because a target didn't work with an early ADC doesn't necessarily mean it's not a viable or valuable target with improvements in ADC design technologies. Importantly, also, most clinically validated targets have multiple active programs in development, where developers are moving forward with innovations designed to improve the outcome for patients, even where an ADC is already approved. So conceptually, we still believe that we can do additional design innovations and make ADCs that work even better. An example of this kind of innovation cycle is that HER2 and TROP2 remain the most studied targets even now, likely due to the high level of clinical validation and an understanding that continued improvements are needed and possible. That all comes back to the power of ADC design. Oops. Here we go. The payload landscape is also a dynamic one. There are over 3,400 disclosed ADCs in development. Leveraging the success of ADCs with tubulin and topo-1 inhibitor payloads with these MOAs still dominate the cytotoxic ADC class. Other DNA targeting payloads and radioimmunoconjugates are also growing in popularity for ADC design choices, and non-cytotoxic payloads are also advancing, with oligonucleotide payloads in particular being an active area. Though tubulin and topo-1 payloads dominate the development space, a variety of payloads have been used in ADCs that have gained approval, as well as a variety of different linker types, ranging from non-cleavable to linkers cleaved by different mechanisms. So while there's a great focus on tubulin inhibitors and topo-1 inhibitors, we have seen DNA alkylators, DNA cross-linkers be incorporated in designs that provided drugs that led to drugs that were approved. But once again, innovation is needed for the next generation ADCs. There are significant limitations with the current and prior generation ADCs that still limit clinical outcome. For example, regarding efficacy, not all payloads work in all indications. A good example of this is tubulin inhibitor payloads in cancers with spindle checkpoint mutations. Those mutations render those cancers very resistant to tubulin inhibitor payloads. Or the emergence of drug resistance can limit the activity of follow-on therapies. Cancers can become resistant to similar MOAs independent of the target use, so new payloads with alternate MOAs are needed. I'll touch on that again. Or potency versus tumor penetration. There was a big push to think about high drug to antibody ratio designs, but it's actually important to match the payload potency and the drug to antibody ratio to achieve antibody doses that are sufficient to drive tumor penetration. And often designs that have payloads that are too potent and too high a DAR don't end up delivering payload well to their tumors. Toxicity still appears to be the major limitation to increasing dose and efficacy. So suggesting that still safety, toxicity, is still a major design consideration when thinking about innovative chemistries and technologies. With regard to toxicity, traditional ADCs are limited by their treatment emergent adverse events. And these common toxicities are cytopenias, neuropathies, GI toxicity, lung toxicity, mucositis, rash, and others. And most ADCs still are dosed near or at their MTD, which suggests that improvements in tolerability would actually lead to improved outcomes. We're not reaching maximal anti-tumor effects, we're reaching maximal tolerated doses instead. So clinical data is now emerging from trials with new generation technologies, such as you'll hear about today from LCB, and continued innovation is still needed to improve these clinical outcomes. One key area for continued innovation is novel payloads. ADC sequencing will become the major development challenge in the future and was a key message at ASCO and ESMO over the past year. By sequencing, I'm referring to what therapies can be used following progression from an earlier line therapy. Much of the concern is that current data suggests that an ADC that follows another ADC with the same MOA shows relatively poor performance and independent of the target used, while good performances are seen when ADCs are sequenced with alternate MOAs. And over the next several years, as the landscape data shows, most indications will have at least one approved ADC with a topo-1 or tubulin payload as a standard of care. Therefore, new payloads with alternate MOAs will be desperately needed. Importantly, emerging novel ADC therapies, such as those that will be presented today, will help mitigate the risk of developing ADCs with new payloads. The improved properties provided by advanced bioconjugation, advanced linkers, and other design elements will contribute to the probability of success for novel payloads. So these technology innovations become even more important as they get applied to new and emerging other technologies. So all aspects of ADC design need to incorporate optimized technologies to continue the march toward better and better therapies. I want to thank you for listening.
Dr. Lutz, thank you. Thank you for your insight looking at this industry. Your Q&A session will be covered after your second talk.
I look forward to it. Thank you.
이어서 스테픈 슬로컴 박사의 리가캠
Next, we
바이오사이언스
will hear
플랫폼
from Dr.
테크놀로지
Stephen Slocum, presenting LigaChemBio's platform technology enables differentiated clinical
Hello, Steve.
strategies.
Good morning.
Thank you for your presentation today. I will now introduce you to the audience in Korean. 슬로컴 박사는 당사의 보스턴 자회사인
Dr. Slocum works at ACB, our Boston-based subsidiary, and serves as the project lead for LigaChemBio's ADC clinical pipelines, including the TROP2 ADC licensed to Janssen and the co-developed B7H4 ADC with NextCure in the US, which is currently being developed. So he's in charge of project development. He is a seasoned expert with over 20 years of experience in oncology drug development.
Please begin your presentation.
안녕하세요 and 감사합니다.
Good morning, ladies and
for taking the
gentlemen.
time,
Thank you very much.
and giving me the opportunity to share updates on the pipeline. So I'll take some time today to discuss LigaChem Biosciences platform technology, enabling differentiated clinical strategies. And in the conversation, we'll address LCB84, LNCB74, and LCB02A. Just an outline of the conversation. We'll start off with a general overview of LCB's technology, as I feel that discussing the core technology and the manner by which it increases preclinical tolerability and efficacy, thereby widening the therapeutic index, is very important to understand the clinical strategies that we've undertaken. So LCB's technology improves patient tolerability and does this through three different aspects. Today, we will talk about two of them. The first is our bioconjugation platform, Conjugol, and the second is our linker technology. A brief overview of Conjugol. This is our bioconjugation system. It's site-specific and homogeneous. So it is achieved by engineering in a small amino acid sequence on the C-terminus of the light chain. This is then specifically functionalized via an enzyme, adding a small inert chemical isoprenoid substrate to the C-terminus, again, of the light chain. This is specific to this particular amino acid sequence at the C-terminus of a protein. So we only see functionalization of this engineered site. This allows, again, for a very site-specific and homogeneous bioconjugation. The isoprenoid is then reacted with our linker payload, which is only chemically compatible with the isoprenoid, maintaining that site specificity and homogeneous approach to bioconjugation. LCB has a number of chemical moieties that will enable either a DAR of 2, 4, 6, or 8, depending on whether or not we add in a mono, di, tri, or tetravalent linker. The linkage itself is plasma stable, unlike previous generation thiol maleimids, which undergo retromichael deconjugation in circulation and lead to a loss of payload. The linker also allows and enables efficient release of the toxin inside of the target cells with tumor-selective proprietary glucuronide trigger. The payload conjugation linker system is compatible to most payloads available, including LCB's proprietary PBD prodrug, which will come up later, microtubule inhibitors, other DNA damaging agents, topoisomerase inhibitors, and payloads that are more exotic. Moving on to our linker, the beta-glucuronide, LigaChem's proprietary beta-glucuronide linker, shorthand is LBG. The mechanism for cleavage and release is shown here. In short, beta-glucuronidase cleaves the sugar moiety within the linker, driving an electron transfer cascade and allowing for the liberation of the parental toxin. This linker itself is plasma stable and tumor labile. LBG is efficiently cleaved by beta-glucuronidase, which is shorthand also referred to as GusB. GusB itself is overexpressed in cancer cell lysosomes, driving efficient release of the parental toxin. The release of the parental toxin itself is important. This enables the native properties of the payload. So in the case of MMAE, this would enable both bystander effect and immunogenic cell death. Other mechanisms of conjugation leave residual atoms on the payload and change those properties. Beta-glucuronidase is active at an acidic pH. pH 6.5 is the optimal pH. This really mitigates any payload cleavage in circulation. So GusB is not commonly found in circulation, but if it were, it would not be active as homeostatic pH is 7.4. Beta-glucuronidase itself has a very interesting expression pattern. There's minimal expression in healthy cells compared to tumor cells, which reduces payload released if there is on-target uptake in off-tumor cells of the ADC. So this linkage both mitigates off and on-target toxicity by minimizing systemic loss and maximizing delivery to tumor cells. Beta-glucuronidase itself is expressed and increased in both the mRNA and protein level across tumor types. This covers both liquid and solid tumors, and we haven't found a tumor type that doesn't express beta-glucuronidase at this point. We believe this expression pattern is due to the inherent metabolic requirements of tumor growth, driving the need for metabolism of any available energy source. And as mentioned, this enables the selective release of the active payload at the tumor itself. As mentioned, this linker is serum stable or stable in circulation. One of the benefits of LCB's technology is that it's often evaluated by external partners. The experiments shown here on the top are data from a large pharma partner who evaluated the technology and bioconjugated the same IgG1, antibody X in this case, with either LCB's bioconjugation system to a DAR4 MMAE or the previous generation val-cit stochastic DAR4 MMAE. You can see across cyno, mouse, and human serum that the LCB ADC, when incubated in vitro, shows no change in DAR over 7 days, whereas the old generation val-cit bioconjugation system shows significant loss of payload even after 24 hours. This result is recapitulated in the cynomolgus monkey, as shown at the bottom, 2 different ADCs showing basically overlapping curves of ADCs and antibodies over time in circulation, demonstrating the lack of loss of payload systemically. The other side of this coin is that LBG releases more payload compared to traditional linkers within tumor cells. So again, these are 2 different large pharma partners who evaluated our technology. On the left, the pharma partner conjugated, again, antibody X with either a val-cit DAR4 stochastic MMAE or a DAR2 MMAE via LBG's bioconjugation system, or a DAR4 with LCB technology. You can see in the red line that an LCB system conjugated ADC with a DAR of 2 releases almost exactly the same amount of payload as a stochastic DAR4 val-cit conjugation. And when you move to the orange line of a DAR4 LCB bioconjugated ADC, it releases considerably more payload within the tumor compared to a val-cit ADC. This is also seen by a separate partner who basically ran the same experiment with a different antibody, looking at the amount of payload released with an LCB linkage in red versus a val-cit linkage in gray. Importantly, the bottom line there is an isotype non-targeting control. So this shows that the LBG system releases more payload on a per mole basis of ADC delivered into the tumor. So this technology here, the LCB linker technology, enables, again, a highly homogeneous ADC with consistent properties. So this abrogates concerns on variable DAR species driving different toxicities. There's no concern of high DAR species causing, severe tox, or low DAR species, not having efficacy. This removes concerns on variable PK between different DARs. Everything is the same that's administered. You can optimize your molecule to drive responses in the tumor rather than drive toxicity systemically. LCB utilizes a stable bioconjugation system. This minimizes systemic exposure of free payload and minimizes potential off-target toxicity due to systemic exposure of payload, and maximizes the payload that's delivered to the tumor. When you think about this, this is very important. We go to all the trouble of making ADCs. You want to have the ADC do its job and take the payload into the tumor and not lose it systemically and then lose efficacy because the antibody is no longer able to drive internalization of the payload into the tumor and cell kill. LBG itself is a tumor-selective cleavable trigger, which increases the delivery of potent parental payloads into tumor cells and mitigates on-target toxicities by a reduction in release of potential payload in healthy normal cells compared to tumor cells. Taken together, this greatly increases the therapeutic index by both increasing safety and potency of LCB ADCs. Moving on into LCB84, our TROP2 targeting DAR4 MMAE ADC, where we have TROP2 targeting ADC with an auristatin payload enabling a differentiated clinical strategy, and LCB's core technology really enabling the use of a potent auristatin payload against TROP2 where others have failed. TROP2 is an antigen, is very attractive, but also very challenging. It's highly expressed in both tumor tissue and in normal tissue, which drives the opportunity to regress tumors, but also drives a lot of on-target toxicity specifically in the skin, esophagus, and oral mucosa. Speaking of the opportunities within the TROP2 space, this is an estimation of market caps across indications where TROP2 has a preclinical rationale or a validated commercial rationale and clinical rationale for efficacy. ADCs have been approved targeting TROP2 in non-small cell lung cancer, breast cancer, and bladder cancer. Drilling down into bladder cancer, TROP2 is expressed across every subtype of breast cancer, HR positive, HER2 negative, HER2 positive, and triple negative breast cancer. Looking at the treatment lines for those three subtypes, HER2 positive breast cancer, you can see second line has trastuzumab deruxtecan, which is in HER2, as Bob has recently mentioned, has a topo-1 payload. And third line has trastuzumab emtansine or Kadcyla, which is a microtubule inhibitor. HR positive, HER2 negative breast cancer also has Enhertu approved in second line, as well as Trodelvy or sacituzumab govitecan. Again, both of those are topo-1 inhibiting payloads. And HR positive, HER2 negative also recently had Dato-A approved in the second line, which is a topo-1 inhibiting payload. And then triple negative breast cancer has sacituzumab govitecan approved in second line or Enhertu approved in second line. So again, a lot of ADCs in breast cancer, but all of them right now, almost all of them have topo-1 inhibiting payloads. So we think that there's a significant opportunity for a non-topo-1 ADC within the breast cancer space. As kind of alluded to earlier, there are a number of emerging challenges in treating breast cancer. And one of the major points of concern is ADC sequencing. When Enhertu was approved, the phase 3 trial demonstrated that you are able to target the same antigen with a different payload class and maintain efficacy. So here, all of the patients treated in DESTINY-Breast02 had already been experienced to Kadcyla, and Enhertu demonstrated excellent efficacy and was approved based on the trial. However, when you start looking at prior exposure to a topo-1 bearing ADC, followed by treatment with a subsequent topo-1 ADC, you see a different story. These are 3 different vignettes at ASCO or ESMO in recent years, demonstrating a drastic reduction in the efficacy of the second ADC bearing the same payload class in sequential treatment. So this, again, really highlights the need for an ADC with a non-topo-1 payload in the breast cancer space, with TROP2 as an attractive antigen to target. It is important to acknowledge that the TROP2 ADC space has a lot of assets in development, but the vast majority of those are topo-1 inhibitors at this point, with Trodelvy and Dato-A both having been recently approved. To the best of my knowledge, the only active tubulin inhibiting ADC under current clinical development is LCB84, really demonstrating the difficulty of driving forward an auristatin or microtubule payload within that space. To really highlight that, we can talk about a clinical vignette on a previous generation TROP2 microtubule inhibitor bearing ADC and its design components. This is RN927, an earlier ADC brought forward into the clinic by Pfizer, targeting TROP2 with a stable conjugation DAR2 oral 101, which is a potent microtubule or inhibiting auristatin derivative. This was linked with a valcit linker with a cathepsin cleavable release mechanism. When you compare this to LCB84, LCB84 has a stable conjugation that's also homogeneous to a DAR4 MMAE with our LBG linker with beta-glucuronidase cleavable release mechanism. When you look preclinically, LCB84 demonstrates increased preclinical tolerability compared to RN927C. Cynomolgus monkey toxicity studies show that LCB84 is considerably better tolerated compared to the Pfizer asset, despite LCB84 having a DAR of 4 versus RN927C having a DAR of 2. It's important to note that both payloads on these ADCs demonstrate comparable in vitro potency. The HNSTD of LCB84 in the DRF study was 12 mg per kg, Q3 week times 2. In the GLP study, the HNSTD was 10 mg per, greater than 10 mg per kg. Q3 week times 2, with histopath findings showing only single cell necrosis at the infusion site. For RN927C, their preclinical research paper states that 6 mg per kg was tolerated in the cynomolgus monkey, with a number of histopath findings covering increased mitoses and single cell necrosis in multiple epithelial tissues in the skin, injection sites, oral mucosa, and esophagus and vagina. In their phase 1 starting dose in the clinic was 0.15 mg per kg every 3 weeks. And when you do the math according to ICH S9 guidelines, that means that their HNSTD in the GLP study was 3 mg per kg. So despite bearing twice the molar amount of payload, LCB84 is greater than 3-fold more tolerated in cynos compared to this asset. When you look at the performance of RN927C in phase 1, it began the clinical trial in August of 2014 and was announced as discontinued in June of 2016. This ADC escalated from 0.15 to 0.3, 0.6, 1.2, and 2.4 mg per kg with no concerns. However, when this ADC was escalated up to 4.8 mg per kg, 50% of patients saw DLTs, including considerable skin toxicity, grade 3 rash, grade 4 toxic epidermal necrolysis, grade 3 bullous dermatitis, and febrile neutropenia. Pfizer then de-escalated twice to try and reach an appropriately safe dose, but was unable to due to continuing incidences of DLTs, further rash, neutropenia, and mucositis. The clinical toxicity profile here mirrors the on-target TROP2 preclinical findings in the monkey, with incidence of skin and oral mucosal toxicities, both reported in the cyno and in the clinic. Importantly, LCB84 does not show significant skin toxicity in cynos at doses equivalent to 3-fold higher than what RN927C was able to be dosed. LCB84 demonstrates a significantly wider preclinical TI, and the phase 1/2 study is actively progressing within the clinic. LCB84 provides a significant clinical and commercial opportunity as TROP2 is a validated antigen target for ADCs. ADCs with a topo-1 payload class play a major role in the treatment landscape of all subtypes of metastatic breast cancer, of which TROP2 is an attractive target for treatment in 2/3 of. Datroway was also recently approved in mutant EGFR non-small cell lung, validating this antigen commercially as a non-small cell lung ADC target as well. Datroway, importantly, is a topo-1 inhibitor payload, so we may be able to enter that space fairly easily. Many additional TROP2 ADCs are being developed. However, the majority of these ADCs utilize topo-inhibiting payloads. ADC sequencing studies demonstrate that payload resistance is a major issue in breast cancer. However, with LCB84, we have the opportunity to sequence with a different payload class and maintain that high efficacy. There is a significant opportunity for non-topo-1 payload inhibitor TROP2 ADCs, with LCB84 demonstrating higher activity and superior tolerability compared to failed first-generation auristatin ADCs. Moving on to LNCB74, our B7H4 targeting DAR4 MMA ADC that's being developed in collaboration with NextCure. This ADC has an auristatin payload that differentiates itself in complicated indication landscapes and utilizes LCB's core technology to drive improved clinical outcomes against B7H4 expression tumors. This is a slide from the most recent ASCO discussing the unmet medical need for B7H4 expressing tumors. The antigen itself is expressed across a number of indications, including breast, ovarian, endometrial, ACC, non-small cell lung, biliary tract cancers. B7H4 has been shown to be a negative prognostic factor, and there's a significant unmet medical need across these tumor types. This is another opportunity to target breast cancer with a non-topo-1 payload. It's also an opportunity to enter into the ovarian space or endometrial space, or to some extent the non-small cell lung. B7H4 as an antigen demonstrates an acceptable level of expression in ovarian cancer. This is data looking across a number of different antigens. When you look at B7H4 in the upper left and compare it to FOLR1, which has successful ADCs developed against it, you can see that it mirrors the same level of expression. Importantly, on the right, B7H4 and folate receptor do not have overlapping expression within tumors. they have exclusivity. So we can target tumors that Elahere is unable to target and not have to worry about following a DM4 payload. There is competition within the B7H4 targeting space. Much of it is topo1 driven at this point, so we don't need to fully be concerned about it, though we are expecting to show efficacy and better safety profiles. What does currently exist, at least in ovarian on the right, is Elahere, which is approved. We don't feel that we have fully overlapping expression profiles, so there are opportunities for treatment there. And Mersana's XMT-1660 or amiltatug ledadotin, which is in phase 1/2 with an auristatin F payload. This is a different representation of that last slide. But we will talk briefly about Seagen's discontinued B7H4V ADC, as well as run a comparison against XMT-1660. This is a schematic of Seagen's B7H4 vedotin. This is a stochastically conjugated average DAR4 ADC with a peptide cleavable linker and an MMAE payload. This ADC has been recently discontinued in phase 1 following Pfizer's acquisition of Seagen. However, this ADC has shown that B7H4 is a viable target for an ADC as they have efficacy data across breast, ovarian, endometrial, and biliary tract cancer. This ADC has a published preclinical cyno-HNSTD of 5 mg/kg in comparison to LNCB74, which has a cyno-GLP HNSTD value of 10 mg/kg and demonstrates a superior preclinical efficacy compared to a biosimilar of Seagen's molecule. That data is shown here. You can see that LNCB74 demonstrates superior efficacy in preclinical models of ovarian and breast cancer when compared to Seagen administered in a single dose. LNCB74 demonstrates regressions and or complete regressions at half the dose of Seagen's molecule. Seagen B7H4V's biosimilar, when administered at 6 mg/kg, really wasn't able to fully regress either tumor, whereas LNCB74 had full regressions at 4.5 mg/kg in ovarian and 3 mg/kg in breast. XMT-1660 is shown here. It's a site-specific conjugated DAR6 ADC utilizing a protease cleavable linker armed with an auristatin analog payload. This payload is advertised to have limited bystander effect. Clinical dose escalation of XMT-1660 began at 0.2 mg/kg, indicating an HNSTD in the GLP study of approximately 4 mg/kg. LNCB74 again utilizes an MMAE payload, so that retains full bystander effect and should enable efficacy in more heterogeneous tumors and potentially drive deeper and longer lasting responses in the clinic. The cyno-GLP HNSTD for LNCB74 was again 10 mg/kg, which is 2.5 times more tolerable than the XMT-1660 molecule. This is the phase 1 dose escalation clinical activity data shared by Mersana at the most recent ASCO. You can see in the subtherapeutic dose range, there is no activity. In the intermediate dose range, there is some but little activity against a number of different tumor types. And in the high dose range of greater than 76.2 mg/m² per cycle, there is an acceptable level of activity. However, at that dose, there is an unacceptable level of toxicity. Notably here, proteinuria, which is a complicated toxicity for physicians to manage, which we think leaves the space considerably open for us to move in and demonstrate better activity and a better safety profile to leapfrog this compound. I am able to speak to some extent about the phase 1 dose escalation of LNCB74. We are rapidly progressing through dose escalation across a number of B7H4 positive indications, including ovarian, breast, endometrial, biliary tract, and non-small cell lung cancer. Our IND was accepted on the 10th of December of 2024, with first patient in 10th of January of 2025. The second dose level was announced as cleared mid-April, and the third dose level was announced as dosing in early May. We have the opportunity and expect to run backfill for both safety and biomarker cohorts to generate a robust clinical proof of concept data set which we expect to be available in the first half of 2026. In summary, B7H4 is overexpressed in many cancers, including ovarian, endometrial, breast, BTC, NSCLC, and ACC. Right now, within that space, Elahere is approved in platinum-resistant ovarian cancer, but there exists ample differentiation between high folate receptor alpha and high B7H4 expression. Most B7H4 and ADCs in development have topo-wide payloads, enabling mechanism differentiation by LNCB74. And within the target indications for B7 overexpression, breast, ovarian, non-small cell lung, and endometrial, there's considerable opportunity for microtubule inhibiting payloads. There are clear and expeditious clinical development paths in existence for LNCB74, as there is significant unmet medical need across these indications. And compared to other microtubule inhibitor-bearing B7H4 targeted ADCs, LNCB74 demonstrates increased activity and a wider therapeutic index. This wider TI is expected to translate to improved clinical outcomes for patients. And importantly, MMAE does not exhibit proteinuria as an adverse event in the clinic, thus de-risking the safety profile of this asset in comparison to XMT-1660. Moving on to LCB02A, our Claudin 18.2 targeting DAR4 topo-1 inhibiting ADC. Here, gastric cancer and other Claudin 18.2 target indications present significant unmet medical need, and LCB's core technology serves to mitigate on-target toxicity and drive responses. Gastric cancer provides a significant clinical opportunity. It is the fifth most prevalent malignancy worldwide. The burden of disease is unevenly distributed, with Asia having the highest incidence, and the global burden of the disease is expected to increase in the next 25 years. So a significant market with a significant number of patients to treat. Claudin 18.2 is expressed at high levels in gastric and GE junction cancers, with additional opportunities in esophageal and pancreatic cancers. Importantly, highlighting the unmet medical need here, advanced gastric cancer has a very poor prognosis with an overall survival of under 1 year. Claudin 18.2 itself is a stomach-specific isoform of the Claudin 18 protein. Its expression is restricted in normal tissue with high levels present in gastric tissue. However, in healthy gastric tissue, Claudin 18.2 is thought to be largely inaccessible within tight junctions between cells. Following transformation, the antigen is mislocalized to the basolateral membrane, becoming accessible to systemically delivered targeted therapies. Approximately 50% of gastric cancer patients have greater than or equal to 2+ IHC staining and greater than or equal to 50% of cells. So generally, if the gastric cancer presents as Claudin 18.2 positive, it is very positive and reasonably homogeneous in terms of target. Importantly, the majority of Claudin 18.2 positive tumors are HER2 negative. This avoids the concern with previous HER2 exposure and topo-1 payload-mediated resistance. Claudin 18.2 expression pattern and intensity is similar across geographies, so the gastric cancer presents the same opportunities across Asia, North America, and other major markets. The estimated market size for Claudin 18.2 indications is shown here, with gastric cancer, GEJ, and pancreatic as the major opportunities. These are the NCCN guidelines for treatment of gastric cancer, both first and second line therapies. Importantly, an anti-Claudin 18.2 antibody, zolbetuximab, in combination with chemo, has recently been approved as first-line therapy in Claudin 18.2 positive tumors, really validating the antigen as a target. But what I think is most important here is that if you are not a HER2 overexpressing gastric cancer or an obvious cancer candidate for immuno-oncology therapy, your opportunities are minimal and are mostly systemic chemotherapy, which really highlights the lack of options in treating this disease and the high unmet medical need. A note about zolbetuximab, the recently approved antibody. Again, this zolbetuximab validates Claudin 18.2 as a targeted therapy target. It's improved in combination with chemo for first-line therapy in Claudin 18.2 positive tumors. However, second-line therapies and beyond are generally lacking. And this, I think, really enables promise to a targeted cytotoxic approach. When you look at the approval data for zolbetuximab, it really provides a minimal PFS benefit. On the left is zolbetuximab plus FOLFOX. On the right is zolbetuximab plus CAPOX. Both of those regimes only give you less than 2 months of additional median progression-free survival. So this is demonstrating really the ability to easily move ahead of zolbetuximab in lines of therapy, given an appropriate level of efficacy. The Claudin 18.2 targeted therapy space is crowded. I won't address the monoclonal or bispecific antibodies here as we are making ADCs. There are some microtubule inhibitor bearing ADCs in later stage clinical development. We don't expect to have concerns following those if needed. The antigen Claudin 18.2 itself is conserved following Claudin 18.2 targeting therapy. For the Topo1 space, SKB315 is in phase 1/2. However, Merck recently returned that asset back to Kelun, and I think that speaks volumes. IBI343 is in phase 2/3, but as far as published data on this asset, seemingly it is in one center in China at this point, so not a lot of clarity around that particular asset. I think also importantly, there is a significant concern of on-target stomach toxicity with this target, which bodes well for LBG's ability to abate toxicities on target. LCB02A is armed with a DAR4 Topo1 inhibiting payload via the LCB platform, bioconjugation, and linker technology. This is expected to provide considerable safety and efficacy advantage versus competitor molecules. LCB02A itself offers an excellent preclinical therapeutic index. There are preclinical PDX models, not CDX models, that show excellent efficacy with an MED of less than 1 mg per kg in tumors. And cyno tolerability is greater than 40 mg per kg with no significant findings. IND-enabling studies are ongoing with an IND scheduled for the first quarter of 2026. And phase 1 clinical studies are planned to be run across Asia and North America in clinical locations with a high incidence of GI cancers. This will enable rapid recruitment and progression through phase 1 in key markets for the disease and generate proof of concept across geographies to build strong justification for advancing and partnering the asset. With that, thank you all for your time.
Dr. Slocum, thank you for your excellent talk. 지금부터 Q&A 시간을 갖도록 하겠습니다.
Now we are going
Q&A는
to start a
당사의
Q&A
최재욱
session.
수업전과 함께 진행하도록 하겠습니다.
Mr. Chejeo
Dr. Slocum, we will now move
participate
to
in this
move on to the
Q&A
Q&A session. Dr. Che will lead. 발표관에 들어온 첫 번째 질문 보도록 하겠습니다.
To look at the first question.
스티브가 답변을 할까요? 제가 할까요? 제가 해도 될 답변일 것 같습니다.
Should I answer or
I'm
Steve
going
will
to take this
answer?
question.
Sure.
질문은 한마디로 지금 요즘 표현으로 중국발 ADC 쓰나미가 몰려오고 있습니다.
So there's a lot of ADCs being developed by China. A lot of trials are happening in China. What are the advantages of LigaChem's platform? It can actually drastically reduce toxicity. China biotech-made ADCs also show great clinical results, according to the question. So can we actually maintain global competitiveness? It's a very critical and practical question. Let me answer this way. Shortly, Dr. Bob Lutz will share our HER2 data. But before that, when you look at the China's clinical outcomes, as was mentioned by Bob, among all the ADCs, regardless of targets, it is if you have to choose, the number one ADC is NR2, but it has the toxicities. It has blood toxicity in 70 to 80 patients, but it's very critical. About 60 to 70% toxicity is acceptable. But when you look at our HER2 ADC's result, grade 2 or 3 is less than 10%. When you look at the overall toxicity of our drug is just half of other competitors. But you're saying China-made ADCs show really great toxicity outcomes. But when you look at all the clinical data, so grade 2, 3, or 4 is less than 10% in our drug, but I didn't see that in other competitor drugs, including China-made ones. So China is actively developing drugs, but there's no innovations in China's ADCs. But they are combining the payloads and conjugation, but they don't have new payloads, no conjugations for their ADCs. So I can dare to say that we are above China in terms of safety and also in terms of innovation. We are ahead of China biotech companies. So regarding platforms, we are technically more above. Thank you very much. Second question. The question was about LCB84's development strategies and concerns about efficacy. Let me answer this way. Regarding Tropto ADC's results are pretty sensitive. LigaChem is Korea's flagship ADC, and it is obtaining a lot of licenses through global clinical trials. LigaChem is working with global partners, so we are following the global standards. With partners and licensees, we have contracts and we follow on those contracts. LigaChem, LEDC, Johnson & Johnson's, Trop2 ADCs. Unless they announce them, we are not supposed to announce them. That's our promise and contract with them. So our official answer is that we cannot answer to your question. But as was addressed by Mr. Steve, preclinical data already proved our excellence. So you can actually assume that even in clinical settings, the excellence will be proven. So our clinical trials are progressing very well, and the clinical trials results will be announced by our licensee, Johnson & Johnson. They will just follow their timeline. So according to the contract, we are not supposed to and we are not allowed to share the results yet.
Jay, I think I understood maybe 2% of that. But I think some of the failings of the Gilead and Daiichi Sankyo TROP2 targeting molecules can be attributed to their linker. Gilead and Trodelvy use an acid hydrolyzable linker where the payload is readily removed from the antibody. So if you're not really able to make a stable bioconjugation and drive payload into the tumor, you'll be able to see regressions and easier lower bars of disease like triple negative breast cancer when you're coming in against standard of care, which is just chemotherapy. But when you're starting to go into other more crowded disease spaces, you'll have trouble driving responses. Datraway also uses a GGFG linker, which is a slower to hydrolyze linker, which would hinder the amount of payload that you can deliver into cells in the tumor. Whereas I think GGFG would have a slower cleavage rate and less payload delivered than Val-Cit. And we've shown that LBG itself will deliver more payload than a Val-Cit on a mole-per-mole basis. So we have a strong rationale for driving more payload into the tumor, giving us a deeper and longer lasting response.
네, 뭐 스티브 답변 아주 간략하게 요약드리면 거랑 사실은 유사한 항암 효과를 나타낼 수 있죠. 그래서 이런 링커의 차별점으로 지금 다른 임상 헐투나 로어롱에서 봤듯이 더 경쟁력 있는 약효를 기대할 수 있을 것 같습니다. 다음 질문.
답변 감사드립니다. QR로 질문 못하신 분 중에서
Thank you very much. If you didn't have a chance to submit your question via the QR code, you can actually raise your hand on site. No question, then let's move on to the next question. Let's look at the second question. So I'm reading the question because it's quite long. I think it overlaps with what Steve already discussed. In a nutshell, if you have toxin A ADC, it's better to use toxin B. Why LigaChem continues with toxin A, although there's already toxin A ADC?
Sure. I just lost the question. That's fine. I think there was a note in there about systemic chemotherapy utilizing topo-1 inhibitors and that being a concern for cross-resistance with a topo-1 inhibiting payload with ADCs. That has interestingly not been the case. There are a number of solid tumors that are treated with topo-1 inhibitors as systemic chemotherapy that do not show resistance to ADCs armed with topo-1 inhibitors. The same is also true for microtubules. There are a number of taxanes used as free payloads and other microtubule inhibiting payloads that don't develop resistance to ADC payloads.
뭐 간단하게 말씀드리면은 그 ADC의 경우에는 페이로드 A 다음에 또 페이로드 A를 사용하는 게 좀 가능성이 떨어지지만 사실은 스몰 모레큘을 A를 쓰고 그 다음에 그와 유사한 페이로드 A를 가진 ADC를 사용하는 경우에는 그렇게 사실은 약효가 떨어지지 않습니다. 뭐 예를 들면은 저희가 이렇게 많이 아는 실제로 병원에서 다들 아시겠지만 first line은 대부분 다 뭐 스탠다드 오브 케어가 키모테라피입니다. 키모테라피에는 당연히 마이크로튜브 이미디터가 들어있는 경우가 되게 많죠. 근데 거기서 약을 조금 투여받다가 잘 안 들으면은 아직도 ADC는 대부분 세컨 라인.
So we are basically looking at the most standard of care. So they are microtubule inhibitors. So when you look at the ORR, so they are not for naive patients. So they already treated patients come back and they are treated with ADCs. There's a payload A ADC, payload B ADC. So which one is better, A or B? This is a big question. In order to make story simple, so we only announce the candidate ADC. But actually, we are producing multiple payloads, A, B, C, D's. Efficacy and toxicities are being studied for them. And based on that, the best therapeutic index ADCs are selected and they are injected as a candidate drugs in clinical trials. We don't just simply go that because there's A, we do B. It's not simple like that. So due to time constraints, Q&A session will be concluded here. So you have 2 QR codes, one for download, 2 for question. But right now, both the QR codes can be used for submitting questions. So after the morning session ends, one of the QR codes will be turned into a download QR code. Now, let's move on to the presentation by Dr. Lutz.
Here is going to talk about the case studies from Exuda Therapeutics of LCB technology in action.
For your second talk today, please begin your presentation.
Great. I will share my screen here.
Yeah, everything's okay now. You can start.
Great. Well, hello again, everyone. I'd like to take you through, hold on. Great. I'd like to take you through 2 case studies of ADCs being developed by Exuda Therapeutics that help to demonstrate the power of LCB's ADC technology. The first program is we call XO14, a HER2 targeting ADC for the treatment of HER2 expressing cancers. And the second program is XO4, a CA242 targeting ADC for GI toxicities. Exuda Therapeutics is an ADC development company headquartered in Newcastle, England, with its development activities directed out of our offices in Boston, Massachusetts. Our most advanced programs are ADCs that contain LCB technology. The emerging data from these programs and others illustrates the power of LCB's technology for improving preclinical and clinical outcomes for efficacy and safety. I'll provide an overview of the findings to date for the XO14 and XO4 programs, and mention some findings for our XO3 program as well that's shown in the table here, a CD19 targeting ADC with LCB technology that led us to the development of XO4. We'll start first with XO14. As I mentioned, this is an anti-HER2 targeting ADC with an MMAF tubulin inhibitor payload for the treatment of HER2-positive cancers, including breast, lung, and GI tumor indications. So a first question to address is why another HER2 targeting ADC? While TDM1 or Kadcyla, a HER2 targeting ADC with a maytansinoid tubulin inhibitor payload, was the first ADC to be approved for the treatment of a solid tumor indication, it was in HER2, or TDXD, as I described earlier, with its high response rates and durations of response that really ignited global interest in the ADC modality. HER2 continues to show impressive response data and is likely to become first-line therapy for patients with metastatic HER2-positive breast cancer, as well as other HER2-expressing cancers. However, there are two major challenges still to face. One is that while TDXD is very effective, it has a challenging toxicity profile, with many patients having to cope with significant GI toxicities during therapy, such as nausea, vomiting, and diarrhea. These toxicities often lead to dose delays and discontinuations. Secondly, the majority of patients ultimately progress during or following TDXD treatment and require additional therapies to control their disease. And emerging clinical data suggests that the best clinical outcomes are achieved when therapies with different mechanisms of action are used in sequence in treatment paradigms for patients. In other words, treatment outcomes have been disappointing when an ADC with a topoisomerase payload is used after another ADC with a topoisomerase payload. So the best outcomes have been seen when sequencing ADCs with alternate MOAs. These findings support the development of a best-in-class HER2 targeting ADC with a non-topoisomerase payload. The relatively low response rate of T-DM1 in second or third line metastatic breast cancer and its lack of sufficient activity in other HER2-positive tumor indications, such as gastric cancer, supported the concept of a next-generation ADC with a tubulin inhibitor payload that is more active than T-DM1, and preferably with a toxicity profile that is better than TDXD. So IXO14 is a novel third-generation HER2 targeting ADC with a modified trastuzumab antibody engineered to be compatible with LCB's site-specific stable conjugate bioconjugation technology. And the ADC incorporates LCB's stable glucuronide cleavable linker with an MMAF auristatin tubulin inhibitor payload. Upon entry into a HER2-positive cell, the linker is cleaved by glucuronidase, releasing the potent payload, leading to cell death. Ixuda has licensed IXO14, now with the generic name of caxmotobart N2-dotin, but I'll keep referring to it as IXO14 for ease, for global development with the exclusion of China and Korea, and is currently conducting a phase 1 clinical trial. Fosun Pharma has licensed IXO14 for development in China under the name FS1502, and Fosun is currently in a phase 3 registration trial for advanced HER2-positive breast cancer in China. One important contribution of LCB's ADC technology in the design of IXO14 is the improvement in the toxicity profile of this MMAF ADC. Traditional MMAF ADCs most of them did not advance into late-stage trials due to problematic toxicities, the most notable one being corneal toxicity. One ADC that has successfully been developed is the BCMA targeting MMAF ADC from GSK called belantamab mafodotin, or Blenrep, for the treatment of multiple myeloma. But comparing the toxicity profile of IXO14 to this traditional MMAF ADC clearly demonstrates the impact of LCB technologies. Drawing from Fosun's phase 1 data presented at ASCO in 2023 on approximately 100 patients at their recommended phase 2 dose, a substantial improvement in the toxicity profile compared to Blenrep was demonstrated. While IXO14 shows excellent activity in its indication, it does so with markedly less thrombocytopenia, GI toxicity as exemplified by cases of nausea, and drastically reduce corneal toxicity and impact on vision. Same payload, much improved tox profile by improved ADC design chemistries. Looking then at IXO14 compared to TDM1 and TXD, we again see the impact of LCB's ADC design innovations on outcomes. While the target and antibody are the same, IXO14 has design changes in bioconjugation with site-specific homogeneous enzyme-based conjugation compared to non-site-specific through lysines or hinge cysteines of the other ADCs. IXO14 uses a low DAR approach to improve tumor penetration, and it has a linker that has improved selectivity between tumor and many normal tissues. It also has minimal bystander, thereby reducing toxicities due to free payload, potentially leading to reduced neuropathies typical of a tubulin inhibitor ADC. The result is a highly active ADC with activity similar to Enhertu in phase 1 patients, but with a notable improvement in its toxicity profile, with lower rates of lung findings, lower GI toxicity, and lower blood cytopenias. This encouraging clinical profile was the driver for Ixuda to initiate their own phase 1 to further explore the dose relationship of IXO14 for efficacy and tolerability. We opened the trial in Australia and have recently expanded to sites in Singapore, New Zealand, and the US. The phase 1 design includes a typical 3+3 dose escalation phase with the addition of enrichment cohorts at selected dose levels. This part 1 of the trial is aimed at identification of the MTD and/or a recommended phase 2 dose, providing sufficient data to allow selection of a dose for a dose expansion phase, or part 2, that will look at specific patient populations, currently anticipated to be HER2-positive breast cancer in patients previously treated with Enhertu, HER2-low breast cancer, HER2-positive gastric and GE junction cancers, and a basket cohort for HER2-positive indications beyond breast, gastric, and GE junction. A snapshot of the preliminary data from Ixuda's phase 1 shows 1, we're observing responses at multiple dose levels, including our first dose level of 40 mg per meter squared. And I'll note here that we're using body surface area dosing that helps normalize ADC exposures across patients with varied body mass indices. And for your guidance, 40 mg per meter squared is comparable to dosing at about 1.1 mg per kg in a patient with a typical BMI. Secondly, we've seen responses in multiple HER2-positive indications, including breast, esophageal, gastric, endometrial, GE junction, lung, ovarian, and gallbladder cancers, with the majority of patients that have shown responses and/or stable disease still on treatment. Thirdly, as use of Enhertu has only recently become common in Australia, we have had only a limited number of post-Enhertu patients thus far. However, recent data has been encouraging, with 4 of 5 post-Enhertu patients showing a response in the higher dose cohorts, and the 5th patient still showing stable disease. The opening of centers in Singapore and the US is intended to increase the number of post-Enhertu patients available for the trial, and we will be presenting a full clinical update at ESMO 2025 in Berlin in October. The safety profile observed so far in the Exuda phase 1 is very consistent with the observations disclosed by Fosun from their clinical experience. The main treatment-related adverse events are generally low-grade, dry eye, dry mouth, and hypokalemia, all of which can be mitigated somewhat by simple interventions such as eye drops, mouth rinses, and potassium supplements. Unlike traditional MMAF ADCs, there has been no high-grade thrombocytopenia or corneal toxicity concerns, and minimal GI or lung toxicity compared to Enhertu. Again, a full update on the safety profile will be presented at ESMO. Overall, Exuda has seen strong phase 1 clinical data with responses seen in post-Enhertu patients and more activity than expected for TDM1 in this population, supporting the potential for IXO14 being an appropriate second or third line treatment option in HER2-positive breast cancer. Additional clinical evaluation will be needed to understand the potential of IXO14 in other HER2-positive indications, but early data is encouraging. And as already noted, IXO14 is well tolerated with a manageable tox profile that is significantly different than ADCs with topo-1 inhibitor payloads. I'd like to switch the focus now to another Exuda asset that utilizes LCB ADC technologies. Our CA24 targeting ADC program grew out of our early experience with another LCB ADC asset, IXO3. IXO3 excuse me, is a CD19 targeting ADC which incorporates LCB's prodrug DNA crosslinker technology. The prodrug approach incorporates LCB's glucuronide trigger chemistry for both the release and full activation of the payload. As exemplified by IXO3, this technology greatly improves the therapeutic index of this highly potent payload compared to prior efforts. The comparator for IXO3 is an ADC developed by Seattle Genetics. SGN CD19B was a CD19 targeting ADC conjugated to a traditional PBD DNA crosslinker called toluene. CD19B used a traditional Val-Cit cathepsin cleavable linker, and development was ultimately discontinued due to poor tolerability. IXO3 with LCB's ADC technology shows comparable preclinical efficacy to CD19B. However, in monkeys, its HNSTD is 5-fold higher. A comparison of the ADC's ability to kill cancer cells in mouse xenografts versus depletion of CD19 positive normal B cells in the monkey provides a window into the power of the glucuronide prodrug format in IXO3. While CD19B depletes normal B cells at a dose even lower than the dose needed to kill cancer cells, IXO3 causes only partial B cell depletion at a dose over 10 times greater than CD19B. This difference is believed to be due to the differential expression of glucuronidase in malignant versus normal B cells, and may be reflective of low glucuronidase expression in many normal tissues. So, with low beta glucuronidase, it's thought that the linker cannot be processed efficiently to cause cell death. IXO3 is currently in a global phase 1 clinical trial for patients with B-cell malignancies. But the data for IXO3 encourage us to ask whether LCB's technology would support use of this payload in solid tumor indications as well. Traditional ADC payloads such as tubulin and topo-1 inhibitor payloads have shown limited activity in hard-to-treat GI cancers. and a DNA cross-linking payload would be expected to be very active in these indications. However, high-potency payloads like PBDs have been limited by poor tumor delivery and poor tolerability. So the improved tolerability of LCB's prodrug approach may make solid tumor targeting a possibility. Given the high potency of the PBD platform, we sought to evaluate the possibility with a highly tumor-selective target, CA242. CA242 is a cancer-associated glycotope with high expression in a broad array of hard-to-treat tumor indications, but has very limited normal tissue expression, suggesting a low risk of on-target toxicity. So IXO4 is comprised of Ixuda's proprietary humanized antibody targeting CA242 with LCB site-specific conjugation, glucuronide trigger and prodrug chemistry, and a highly potent PBD DNA cross-linker. The ADC shows highly potent, target-dependent in vitro tumor cell killing, with the in vitro potency correlating well with target expression levels, with very low EC50s observed with cells expressing moderate to high levels of CA242. The ADC also exhibits strong bystander activity, where antigen-negative cells can be depleted in the presence of a low percentage of positive cells. No activity is observed with the negative cells in the absence of target positive cells, and this bystander activity can potentially improve the activity of the ADC in heterogeneous CA242 expressing tumors. IXO4 also shows strong in vitro activity across a range of models. However, initial studies were a bit surprising in that higher expression did not result in increased in vivo efficacy. In fact, the highest expressing models had the higher minimally effective doses. This inverse correlation is indicative of an antigen barrier effect. This effect is due to high target expression limiting the penetration of the ADC into the tumor. Rather, the ADC is captured on tumor cells that are closest to the blood vessels, as shown by the middle figure, the illustration on the left. This antigen barrier challenge is particularly important for ADCs with high potency payloads, as the high potency can limit the clinical dose of the ADC. This necessitates another approach to drive penetration. Investigation by Greg Thurber's laboratory at the University of Michigan and others have demonstrated that ultra-low DAR regimens by co-administration of the ADC with unconjugated antibody can help overcome the antigen barrier effect and improve ADC distribution within the tumor, and thereby increasing efficacy. This is particularly useful for antigens with high expression. And the immunofluorescence shown on the right clearly demonstrates the improved penetration and overall delivery with increased antibody dose. Evaluation of the efficacy of this ultra-low DAR approach with IXO14 shows how impactful it can be. In the study shown on this slide, with a high-expressing gastric cancer model, the original study showed that the MED, the minimally effective dose of IXO4 in this model, was 1 mg per kg single dose. Co-administration with 10 to 50-fold excess of unconjugated antibody improved the MED down to 0.4 mg per kg. This approach may not provide improvement for all targets or for all levels of target expression, though preclinical data has shown improvements in efficacy for a wide range of targets and may be an appropriate approach for many applications. For CA242, since the majority of patients since the majority of patients that are CA242 positive have moderate to high expression, as exemplified by our models, we've chosen to take this ultra-low DAR regimen into the clinic. You see the percentage of patients that have moderate to high expression in the graph on the left. All of the blue patients would be those considered for this type of approach. About 80% of colorectal cancer, GE junction, gallbladder, cholangiocarcinoma, and pancreatic cancers show moderate to high expression of CA242. In gastric cancer, it's about 60% of patients that have high to moderate expression. This leads to a very large potential patient population that have failed frontline chemotherapies with this level of CA242 expression. Estimates of greater than 2 million patients may be expected to need second line or third line therapies annually that would have high to moderate CA242 expression. Interestingly, this ultra-low DAR approach is not dissimilar to what's being evaluated for radioimmunoconjugates, and the FDA has provided positive feedback to Ixuda regarding using this ultra-low DAR regimen for IXO4 clinical evaluation. IND studies are well underway, with a US FDA IND filing expected later this year. Our trial design will consist of a dose escalation phase, including enrichment cohorts at select dose levels, followed by a dose expansion phase to look more closely at selected indications, along with a basket cohort, very similar to what is planned for IXO14. And we're incredibly excited to bring this program into the clinic. Finally, I'd like to thank all the patients, their families, caregivers, and investigators for their participation in our clinical trials, and also the Ixuda and LCB teams for their efforts to support these programs. I thank you for listening and will be happy to take any questions.
So 14 clinical data.
The question is not yet on my screen.
Okay, so let me read this for you, and then.
Now I see it.
Okay, probably this is the question you can answer.
Do you think? Yes. Even the experience so far with TDM1 suggests that prior therapy with Enhertu does not decrease the response rate to ADCs with a different payload, in the case of TDM1, a tubulin inhibitor payload. So our initial target product profile is to position IXO14 post Enhertu, and essentially go head to head with TDM1 or other HER2 targeting small molecule agents. I think there's a high potential, given what we've seen and the response rates, at least in phase 1, that are similar to Enhertu, and we'll know some about Fosun's phase 3 data over time, that IXO14 may also be competitive with current first-line standard of care, namely trastuzumab and taxanes. So I think we have an expectation that IXO14 could be positioned second line post Enhertu. Now, direct competition with Enhertu would be challenging, although it may be possible that a large strategic partner would be interested in taking on that role. But the high response rates for Enhertu do provide somewhat of a challenge to think about direct competition. But again, I think we're well positioned for second or third line therapy. I would say currently no other ADC has as favorable a response and toxicity profile with a payload other than Topo 1.
Okay, thank you, Bob. Let me just translate your answer to Korean just for the next couple of minutes. 질문은 사실 오늘 발표 중에 뭐 제일 중요한 것 데이터인 것 같은데 NR2를 맞은 질문은 그러면은 이 리가캠의 익수다의 그 헐투 ADC를 과연 이처럼 NR2 트리트먼트 된 다음에 포스트 NR2로 할 것이냐 아니면은 헤드 투 헤드로 컴피티션을 할 것이냐 뭐 심플한 질문인데 사실은 굉장히 여러 가지 팩터들이 포함된 사실 답변을 드릴 수밖에 없는데요. 이제 밥의 앤서는 아까 밥이 설명했듯이 원래 마이크로튜블 이니비터를 포함하는 TDM1을 맞고 잘 안 듣는 환자들이 그 토포아이 페이로드를 가진 NR2에 의해서 치료 효과가 잘 나타났듯이 아마 비슷할 것이다. 실제로 이제 4명의 PR이 나왔으니까 그 입증은 됐죠. 그래서 NR2 다음에 우리 헐2가 들어가서 그건 충분히 경쟁력이 있다. 그래서 현실적으로는 지금 이제 임상에 들어가거나 혹은 허가를 받아도 이미 사실은 NR2를 맞은 환자들이 엄청나게 많을 거예요. 그러니까 첫 번째로 나이브한 환자들이 저희한테 오지는 않을 거니까. 그러니까 세컨 라인으로 포스트 NR2는 너무나 당연하게 차지할 것이다. 근데 이게 정말로 모든 회사들이 지금 찾고 있는 솔루션이거든요. NR2가 점점 더 매출도 많이 오르고 이제 인기 있어지면서 과연 그러면은 NR2 안 듣는 환자들은 어떻게 할 거냐? 그냥 터미널로 끝날 것이냐? 솔루션이 없는 상태에서 저희처럼 사실은 이런 임상 대회를 통해서 포스트 NR2가 솔루션이 있으면 굉장히 좋죠. 그다음에 헤드 투 헤드 컴패리슨은 초기 단계에서는 사실은 챌린징 할 수도 있지만 익수다가 그 퓨처 뭐 파트너십 얘기를 잠깐 했는데 뭐 끝까지 사실은 나중에 막 대규모 삼상 같은 거를 뭐 할 수도 있겠지만 하시기는 현실적으로 힘들 테니까 만약에 미래 빅파마 파트너가 테이크오버를 해서 그런 거에 인트레스트 한다면은 실제로는 컴패리슨 스터디도 뭐 가능하겠죠. 왜냐하면은 아까 보여드린 대로 NR2는 여전히 굉장히 많은 부작용 때문에 환자들이 사실은 그 서퍼링하는 부분들이 있는데 여태까지 정말로 보여주지 않았던 아까 보셨다시피 뭐 구토나 뭐 모든 면에서 사실은 혈구 독성이라는 것에서 현격하게 낮기 때문에 그런 측면을 생각하면은 나중에 미래 뭐 빅파마라고 얘기하지는 않았지만 그런 파트너가 헤드 투 헤드의 임상 디자인도 가능은 할 것이다. 이렇게 대답했습니다. 네.
예, 오늘은 데이터가 좀 짧게 나왔지만 자료에 띄웠듯이 에스모에서 한 번 더 발표할 예정입니다.
So there will be another presentation at ESMO. So detailed data will be presented at ESMO. We'd like to move on to the second question.
Bob, the second question is coming up.
Yep. I'm reading through it. Very
Okay.
interesting. Okay. Very, very interesting question. So I do believe that the payload plays the biggest role in and the ability to utilize that payload, as in the case of IXO14, since a traditional MMAF conjugate would probably not have been sufficient. It's payload MOA that I think is the biggest driver of next favorable outcome, more than the antibody or the data thus far, in large part driven by studies post Enhertu, have suggested that following with the same payload, regardless of target, shows poor performance. Deciding whether to follow with HER2 or a TROP2 ADC with a different payload, such as IXO14 versus LCB84, for example, is a question that's difficult to answer at the moment.
But it doesn't need to be 84. It can be like a Trodelvy or like a Daiichi Sankyo's
Well,
TROP2.
yeah. So any topo 1 payload after a topo 1 payload, the current data suggests that will show poor performance, regardless of target. So payload's the most important factor thus far in our understanding of how best to sequence ADCs.
Great. 이 질문도 사실은 굉장히 중요한 질문인데요. NR2에 대한 내성이 한마디로 항체가 중요하냐, 페일로드가 중요하냐 그 질문입니다. 그러니까 토포 I에 대한 내성이 암세포에서 생길 수도 있고요. 혹은 헐투 엔티젠 자체가 뭐 돌연변이거나 없어져 버리면 사실은 그 다음에 헐투 ADC를 처리해도 또 계속해서 내성이 지속될 거니까. 근데 지금까지 임상 데이터들을 종합해 보면 지금까지 밥의 질문도 그렇고 전체적인 컨센서스는 페이로드가 더 중요하다입니다. 그 내성에 있어서. 그러니까 뭐 예를 들면은 100명의 환자가 HER2 ADC의 내성을 보이는데 그 중에 페이로드 내성이 더 많은지 아니면 HER2 자체가 뭐 익스프레션이 변하거나 어떤 게 더 많냐 보면은 실제로 페이로드 내성이 더 많다는 게 지금까지 임상의 데이터고요. 그래서 여기 보면은 만약에 HER2가 변한 거면은 트롭2 같은 거 트롭2 ADC로 그냥 처리하면은 그게 극복되지 않겠냐 사실은 질문에 그게 포함이 돼 있는데 밥의 앤서도 그렇고 지금까지 임상 데이터로 예를 들어서 내성 환자를 일일이 다 검사를 해서 이게 헐투가 바뀐 건지 아니면은 토포아이의 내성이 있는지 그거를 일일이 다 뭐 유전체 분석을 하거나 이래서 그 다음 단계의 약물의 임상에 사실은 들어가는 경우는 매우 드뭅니다. 그냥 안 들으면 NR2가 안 들으면 사실은 그 다음 약물로 진행되는 경우가 많거든요. 그래서 그런 경우에 지금까지 임상 데이터를 보면은 트로델비나 그러니까 NR2가 안 드는 환자를 트로델비나 트롭2 ADC로 처리를 해보면 반응성이 되게 안 좋았어요. 그 얘기는 토포아이가 들어간 ADC를 한번 한 환자들은 그 다음에 타겟은 다르더라도 똑같은 페이로드인 토포아이가 있는 경우는 사실은 잘 안 들은 경우가 많습니다. 그래서 이거에 대한 답은 페이로드를 사실은 바꾼 ADC로 트리트하는 것이 사실은 가장 좋다가 지금까지의 통계학적인 답변입니다.
예, 세 번째 질문 올려주십시오.
So we'd like to move on to the third question.
그러니까 흰 글씨가 좀 잘 안 보이는 것 같아서 검은 글씨로
So
바꿨습니다.
we have changed the colors to make it more remarkable. Can you please take a look at the third question on screen?
Hey Bob, do you see the
Another great question. So one of the things that's emerging about HER2 is that there are very few patients that are actually HER2 negative. Although it is clear that low and ultra low activity of HER2 is probably driven by a lot of tumor microenvironment release of the topoisomerase payload. That being said, we are with IXO14, Ixuda and Fosun have seen activity in HER2 low. I wouldn't say we have experience with ultra low. So it's not clear that the post and HER2 market for compound like IXO14 would only be current HER2 positive high breast cancer. On the other hand, it is going to be important for ADC development in the future for ADCs to carve out more and more specific patient characteristics that will help differentiate from other next generation ADCs that are coming along with targets in certain populations and so forth. So, for example, post and HER2 market for HER2 breast cancer, HER2 high breast cancer, and potentially HER2 high gastric and esophageal cancer will be a higher than $1 billion market in itself. So I think not a bad market for a second or third line therapy in that case. The plus side of MMAF not having bystander effect is it's less cell permeable. So in a target directed cell, in a target positive cell, it can accumulate to eventually the concentrations that are needed to kill the cell. So that in part compensates for lack of bystander activity. Is that you're not connected, you're not dependent on the kinetics of delivery, that you can take more time to deliver payload through with lower antigen density. The upside, of course, is incredibly important for tubulin inhibitors. One of the biggest clinical relevant toxicities of MMAE conjugates, which you don't talk about often because it's not part of the DLT window, is neuropathy. And when patients are treated with a vedotin ADC for multiple cycles, neuropathy is often an important clinical toxicity that has to be dealt with. MMAF is expected to show very limited neuropathy because the free drug is not permeable to nerve cells. So it is a balance.
Okay, great. 질문부터 뭐 잠깐 설명드리면 뭐 다들 이해하셨겠지만 MMAF는 전통적으로 굉장히 소수성이 높다고 인식되고 있어서 암세포에서 암세포를 죽인 후에 옆에 세포를 잘 못 들어가요. 그래서 이제 바이스탠더 이펙트가 적다고 하는데 이렇게 적으면 한마디로 HER2 익스프레션이 굉장히 나는 캔서에서는 옆에 암세포를 죽이는 거에 좀 제한적이니까 이렇게 HER2 low나 ultra low breast cancer에서는 한마디로 LCB의 ADC는 잘 안 듣지 않겠냐 하는 게 뭐 이 질문의 일단은 요지고, 그렇게 되면은 포스트 NR2 마켓에서 HER2가 높은 환자에 사용된 포스트 NR2 환자에만 되지 않겠냐, 그래서 시장이 좀 줄어들지 않겠냐인데 밥이 조금 길게 설명을 했는데, 일단은 그동안 호주에서 진행된 임상에서 실제로 HER2 로도 포함이 돼 있었어요. 근데 HER2 로에서도 저희가 효과가 잘 나오고 있기 때문에 실제로는 아마도 포스트 NR2에서도 HER2 로에서도 저희가 뭐 기대할 수 있을 겁니다. 그래서 뒷부분에 설명한 거를 뭐 밥이 설명한 걸 앞부분으로 당겨오면 바이스탠더 이펙트가 없다는 게 굉장히 단점 같지만 일단은 독성의 장점이 있고요. 왜냐하면 옆으로 퍼지다 보면 결국은 노멀 셀도 죽이게 되니까. 두 번째로 바이스탠더 이펙트가 없다는 것의 장점은 뭔가 하면은 왔다 갔다 셀 밖으로 안 하기 때문에 MMAF는 축적이 많이 됩니다. 그래서 한마디로 블러드에서 ADC가 서큘레이션이 오래되면은 들어온 게 안 나가요. 예를 들어서 MMA 같은 거는 왔다 갔다 하고 나가게 되거든요. 그래서 뭐 같은 오리스테틴이라도 장단점이 있다고 이제 밥이 설명한 이유는 MMA ADC는 쓰면 항상 뉴로패티라는 게 나와요. 그러니까 신경계로 굉장히 아픔을 호소하는 신경 말단 고통을 호소하는데 그 이유가 신경세포로 MMAE가 쑥쑥 잘 들어가서입니다. 그러니까 떨어져 나오는 프리 페일로드가. 근데 MMAF는 그런 게 없어요. 왜냐하면 바이스탠더드 이펙트가 없으니까. 떨어져 나온 게 못 들어가죠. 대신에 ADC로는 한 번 들어가면은 안 나오고 그 다음에 ADC가 또 들어오고 하니까 바이스탠더 이펙트가 없다는 게 그냥 무조건 효능이 더 좋지 않음을 의미하지는 않습니다. 지금 밥이 설명한 대로 이런 이제 어큐뮬레이션이 되기 때문에. 그래서 결론적으로 말씀드리면은 헐투로도 저희가 충분히 가능성이 있다고 생각하고 아마 포스트 NR2 마켓에서 무조건 헐투 하이로 국한되지는 않을 것이다. 근데 플러스 이제 밥이 얘기한 것 중에 하나 또 중요한 포인트는 헐투 하이 브레스트 캔서 게스트릭만 합쳐도 그거만 합쳐도 $1 빌리언 시장이 넘으니까 아주 보수적으로 예를 들어서 정말로 헐투로를 뭐 우리가 많이 못 먹는다 하더라도 헐투 하이만 해도 충분히 큰 시장이기 때문에 우리 헐투가 충분히 뭐 경쟁력이 있을 것이다. 근데 그건 아주 보수적인 에스티메이션이고, 그러니까 헐투로에서도 지금 효능이 나오고 있기 때문에 헐투로까지 진출이 가능할 것입니다.
예, 발표 시간상 질문 하나만 더 보고 가겠습니다. 질문.
Due to time constraint, we are going to take the last question because time is running out.
뭐 특정 ADC랑 비교하는 거라서 뭐 저희가 정말로 풀 데이터셋을 이렇게 쫙
This is
테이블화해서
compared to specific
정말로
ADCs. So, for example, we can present a full table showing what would be the safety and toxicity. Without that full table, it would be very difficult to answer this question. So it looks similar, but what are the advantages? So we really need to have very sophisticated data.
the clinical outcome data set from this the another HER2 ADC. It's probably not too easy to answer this question, but you can always add your comments on it.
Well, one of the things I would say is we currently wouldn't consider ADCs with topo-1 isomerase payloads as competitors for the treatment space that we're aiming for. So neither of these, I think both of these ADCs that are mentioned in the question have topo-1 inhibitors, and they will likely suffer in the post and HER2 setting when that becomes relevant in China.
That's a great point. Yeah.
Where and that was part of the concept of developing another HER2 ADC with using the LCB technology, but with a potent tubulin inhibitor like MMAF. Our target market, our competitors are those with non-topo-1 inhibitor payloads. And right now, the clinical data from Fosun and emerging from Exuda suggests this IXO14 has the potential for best-in-class in non-topoisomerase payload ADCs.
네, 지금 밥에 포스트 NR2 환자를 만날 텐데 여태까지 다른 임상에서 보듯이 토포아이를 맞은 사실은 환자들이 들어왔을 때 또 다른 토포아이 ADC가 효능을 보일 가능성은 사실은 매우 낮기 때문입니다. 그래서 이거를 제가 부연해서 설명드리면 이런 헤드투헤드 정말로 굉장히 큰 환자군에서의 데이터셋은 정말로 둘 다 허가를 받았으면 의미가 있는데 뭐 달리 말씀드리면 저희 HER2와 이 HER2 ADC가 그런 포스트 마케팅에 비교가 될 가능성이 거의 없어요. 왜냐하면 거기까지 가기 전에 저희가 이 ADC건 저희 ADC건 임상을 2상을 3상을 거듭하면서 저희가 만나게 될 환자들은 first line이 아니기 때문에 이미 키모도 맞고 NR2도 맞고 사실은 이런 환자들을 맞닥뜨리게 되고 그 환자들을 대상으로 저희가 ORR 반응성이나 독성 같은 것을 전부 다 이제 계산해서 나오기 때문에 또 다른 토포아이 ADC가 들어가서 그런 포스트 NR2 환자를 만나서 굉장히 좋은 데이터가 나올 가능성은 사실은 높지는 않습니다. 그러니까 다른 캔서 같은 경우는 예를 들어서 CRC 같은 경우는 뭐 예를 들어 보면은 ADC가 많이 안 들어가 있으니까 ADC를 맞은 환자가 있을 가능성이 매우 낮아요. 근데 브레스트 캔서 같은 걸 생각을 해보면은 요즘 당연히 사실은 NR2나 TDM1이나 헐투 기반으로 한 ADC에 대해서 하는 경우가 많으니까 현실적으로 그렇게 될 것 같습니다.
예, 이번 시간 Q&A 세션은 마치도록 하고.
This concludes Q&A session with Dr. Bob Lutz. And then today there was a media that saying that there will be a clinical trial expansion about how to post. And some people say that the QR code is wrong, so we have corrected the QR code. You can find it outside. Thank you.
Now, the last presentation by Dr. Chingmei Shi. She's going to make a presentation about CS5001, Global Clinical Trial Progress.
Presentation today. I will now introduce you to the audience in Korean. 예, 시 박사님은 시스톤 파마스티컬의 치프
Dr.
메디컬
C
오피서.
is a chief medical officer of Stone Pharmaceuticals. She brings over 20 years of experience in the clinical and pharmaceutical industries with deep expertise in oncology and hematology. Let us now begin the presentation.
Thank you. Let me share my presentation slide. Can you see my slide deck?
Not yet. Hold on a second, please.
Sorry.
We are working on it. Please hold on a second, please.
Can you see it now?
Not yet.
Okay.
We are working on it. Please hold on a second. Dr. Shi.
Sure.
Okay. 잘못 올린 것인가? 잘못 올린 거예요? 우리가 올릴 거예요? 우리가 올릴 거야? Everything's okay now. You can start.
Okay, thank you. Good morning, everyone. My name is Chingmei Shi. I'm the Chief Medical Officer from Stone Pharmaceuticals. We are a China biotech company. It's my pleasure today to have the opportunity to introduce CS5001, Stone's global competitive ROR1 targeting antibody drug conjugate. With best-in-class potential and rapid clinical development program, We believe CS501 is a cornerstone of our innovative pipeline 2.0 strategy. Let's dive into why this asset is positioned to capture significant value in oncology and in system. A brief introduction about System, which is our company. System Pharmaceuticals is an innovative biotech company driven by global recognized research and development capabilities. We have established an efficient and high-quality clinical development engine. As of 2025, the company boasts 16 NDA approvals with over 50 data presentation publications and over 10 ongoing discovery projects, alongside over 40 IND approvals. While leveraging a clinical insight-driven modular research and development model, we have a proven track record in high-quality drug development worldwide, with strategic partnership such as collaboration with MNCs like Pfizer to accelerate our commercialization process. Currently, we have 4 commercialized products covering 9 indications across 5 territories. With sugammalimab, which is an anti-PD-L1 antibody approved in China, EU, and UK, demonstrating our strong global momentum. Our strategy centers on 3 pillars. First is a commercial stage program, as I mentioned earlier. This includes sugammalimab, a PD-L1 antibody, and pralsetinib, which are RET inhibitor, and avapritinib, which is a KIT PDGFR alpha inhibitor. And second pillar is our pipeline 2.0, which target high potential novel therapies, including CS501, a ROR1 ADC. This ADC is a global number 2 contender with best-in-class potential, and PD-1, VEGF, and CTLA-4 trispecific antibody. And the third pillar is our early strategy innovations, aims to secure long-term leadership. This includes bispecific antibodies and next-generation ADCs. CS501 anchors our ADC portfolio. This is a compound with global rights, excluding South Korea, and first-to-market potential. This compound targets both solid tumor and hematological malignancies. Currently, we have phase 1b study ongoing, with registration pathways accelerating. Let's dive into CS501 development program. First, let's look into the design of the compound. CS501 is not just another ADC. It's engineered for superiority through 4 key innovations. First is a full human anti-ROR1 antibody for minimal immunogenicity with DAR to homogeneity while site-specific conjugation. In addition, it has tumor-selective linker, which is stable in serum, but cleavage by tumor enzyme beta-glucan. And the payload is a PBD dimer prodrug, which can be activated only in tumors. This delivers high potency and low systemic toxicities, comparing with other MMAE or DXd-based ADCs. PBD has stronger killing of slow-growing tumors while DNA cross-linker mechanisms and reduced likelihood of inducing tumor resistance. So why we target ROR1? As we know, ROR1 is overexpressed in solid tumors like triple-negative breast cancer, like lung cancer, ovarian, etc., as well as hematological malignancies like diffuse large B-cell lymphoma like CLL, etc. but the expression is almost absent in most healthy tissues. And Cystone's proprietary MAB antibody has validated RR1's expression via immunohistochemistry, as illustrated in the figure on the right-hand side. In the solid tumors, the expression is around 50 to 90%. Let's look at the preclinical data. CX-501 design translates to exceptional efficacy in preclinical studies. As shown in this figure, CX-501 exhibits high affinity to human binding to selectively to RR1, but not RR2. In tumor cells, on the right-hand side, it shows in tumor cells, CX-501 demonstrates rapid and deep internalization at 37 degrees Celsius. This facilitates toxin release and action. CX-501 also shows potent ROR1-dependent cytotoxicity in solid tumor and hematological malignancy cell lines. For example, in JAK1 cells, which is a mantle cell lymphoma cell line, CX-501 has an IC50 of around 1 nanomole versus around 100 nanomole for iso-ADC, representing a 90-fold activity difference. In tumor xenograft models, a single dose of CX-501 at 1 milligram per kilogram achieved 109% tumor growth inhibition, with 2 out of 8 complete responses. This outperforms the benchmark ADC, even at higher dose level. CX-501 demonstrates robust bystander killing in co-culture assay with ROR1 positive and ROR1 negative cells, eliminating tumor cells adjacent to ROR1 expressing cells. This is important considering the heterogeneity feature of tumor tissues. In patient-derived xenograft models of solid tumors, CX-501 achieved 104% tumor growth index, tumor growth inhibition in ROR1 high tumors, compared to 53% for iso-ADC. This validated CX-501's efficacy in real-world tumor context. I want to share some of the clinical data with you. CX-501's clinical development targets both lymphomas and solid tumors and spans first line as well as later line settings as monotherapy and in combination. Phase 1A is a monotherapy dose escalation study, while Phase 1B is a dose expansion study with pivotal extension potential. As for lymphomas, we target in diffuse large B-cell lymphoma. This includes later line monotherapy. This includes monotherapy in relapse and refractory setting, first line in combination with R-CHOP, second or third line in combination with R-GemOx and R2. In addition to DLBCL, we are also exploring indolent lymphomas such as follicular lymphoma, marginal zone, CLL, etc. As for solid tumor, we are exploring CX-501 as monotherapy in RR1 positive solid tumors and in combination with sugammalimab, which is our anti-PD-L1 antibody in all-comer solid tumors. Phase 1A data validated CX-501's promise. data disclosed at ASH 2024 showed CX-5001 is well tolerated in heavily pretreated B-cell lymphoma patients. As data cutoff for September 2024, we have 33 advanced B-cell lymphoma patients treated across 6 dose levels, starting from 33.5 to 156 microgram per kilogram. Of the 33 patients treated, more than 2/3 of patients have performance status of 1, and 82% of patients received at least 3 lines of prior treatments regimens. 12% of patients received prior CAR T-cell therapy, and 9% patients received prior transplant. For safety, we have tested 10 dose levels up to 195 microgram per kilogram. We did not identify any DLTs in this heavily pretreated patients group. Safety profile is overall manageable. We have around 46% of patients with grade 3 or higher treatment-related adverse events, with most common ones being hematological toxicities such as anemia, white blood cell count decrease, and liver toxicity such as AST increase. We didn't identify unmanageable neurotoxicities unlike MMAE competitors. Recommended phase 2 dose is currently tentatively set as 125 microgram per kilogram for lymphoma, which is dose level 8. In the dose escalation phase, we have observed preliminary efficacy in Hodgkin's lymphoma. Among 10 available patients at dose level 5 to 9, overall response rate was 60%, with 100% ORR at dose level 8, which is our tentative recommended phase 2 dose. This includes 2 CRs and 1 PR. As for non-Hodgkin's lymphoma, among 16 available patients at dose level 7 to 9, overall response rate was 56%, with 70% ORR at dose level 8. This includes responses in both aggressive B-cell lymphoma such as diffuse large B-cell, mantle cell, and high-grade B-cell lymphomas, as well as indolent lymphomas such as marginal zone and follicular.
Dr. Shi, sorry for
data
interrupting.
suggest-
Dr. Shi, so we got the
Yes.
Yes, sound is a little winging. So could you mic little away from your mouth, please?
You mean my voice is too weak?
Yeah, it is okay. It is okay now.
Okay, thank you. So let me continue. So preliminary efficacy data suggest CX-5001 monotherapy demonstrate a higher response rate than our competitor, which is CLOV, in aggressive lymphomas, over 50% versus around 30% in diffuse large B-cell. And we also, with CX-5001, shows activity in indolent lymphoma also. As for solid tumor, CX-5001 demonstrates anti-tumor activity in solid tumor with partial responses and stable diseases observed in non-small cell lung cancer, pancreatic, triple-negative B-cell lymphoma, triple-negative breast cancer, and ovarian cancer at higher doses. Notable result includes one partial response in non-small cell and pancreatic cancer respectively, and stable diseases in lung cancer, in breast cancer, as well as ovarian cancer. This highlights CX-501's potential beyond lymphomas. As for PK profile, CX-501 exhibits dose proportional drug exposure with half-life around 5 days. There's no significant accumulation of free toxin detected. This confirms the linker stability in the serum. Let's move to the commercial potential of CX-501. As we know, CX-501 stands as one of the 2 leading ROR1 ADCs globally, competing with Merck's Zilowski. The key commercial advantages include of course, the first is clinical differentiation. For diffuse large B-cell lymphoma, CX-501 has overall response rate of around 50%, while at tentative recommended phase 2 dose response is around 70%, while the Zilowski has shown around 30% of response rate. In addition, we have broader indications, including indolent lymphoma, as well as solid tumors. As for safety, as I mentioned, we don't have any DLT, dose limiting toxicity reported up to dose level 10. And at ASH 2024, actually Merck presented data for Zilowski in combination with R-CHP as first-line treatment for patients with diffuse large B-cell lymphoma. And utilizing a reduced dose of Zilowski, the combination regimen demonstrates a near 100% durable complete response rate, which is quite encouraging for ROR1 targeted therapy. In addition, Lonca, which is a CD19 PBD ADC, data also supports development of combination therapy with PBD-based ADCs. As shown in this slide, Lonca in combination with rituximab in relapse and refractory DLBCL achieved ORR of 80% and a complete response rate of 50%. With no new safety signals. In addition, Lonca in combination with glofitamab, which is a CD20, CD3 bispecific T-cell engager, demonstrates around 100% overall response rate and 78% of CR rate, with a well-tolerated safety profile. So these 2 competitors' data actually give us quite promising development strategies for us to move to first-line and later-line combination treatment in diffuse large B-cell lymphomas. As for the global hematologic malignancy market, this is expected to reach $139 billion by 2029. With diffuse large B-cell lymphoma representing the largest indication, which is around 31% of incidence. And solid tumor markets as parallel value, given CX-501's activities in solid tumor like non-small cell lung cancer and pancreatic cancers. And building on CX-501's demonstrated activity and emerging competitor data in lymphoma, such as diffuse large B-cell lymphoma, we are advancing our targeted registration strategies. For phase 1b expansion, we are actually evaluating the monotherapy in relapse refractory diffuse large B-cell lymphomas with a potential of pivotal single-arm expansion. This has already been initiated. And for combination studies, for first line, well, of course, we are evaluating the for first line diffuse large B-cell lymphoma, we are evaluating the CX-501 plus R-CHOP. And for second, third line, we are evaluating CX-501 plus R-GemOx or R2. So in summary, CX-501 represents a best-in-class potential while optimized ADC design, cleavable linker, non-cleavable linker in the serum, as well as a product design. We have higher response rate in aggressive lymphoma as well as in indolent lymphomas. And this is the first ROR1 ADC has shown promising anti-tumor activity in solid tumors. CX-501 demonstrated a favorable safety profile, which we believe will enable our combination strategy. And currently, as I mentioned, phase 1B study is ongoing for dose optimization for monotherapy in RNR setting and with potential single-arm registration. And the first line and second, third line DLBCL, combination DLBCL, and mono and combination therapy in other B-cell lymphomas, as well as solid tumor expansions. We are actually in the process of finding global partnership to optimize CX-501's value. We believe its dual hematologic and solid tumor promise. CX-501 is positioned to transform outcomes for patients and will drive significant value for this compound as well as for System and LigaChem. With this, and this ends my presentation. Thank you.
Yeah, Dr. Shi, thank you for your great talk. Join and lead. 발표가 나온 질문 한번 살펴보겠습니다. 첫 번째 질문
Let's
올려주십시오.
read the first question. Would you please load the first question on the
Hey, Dr.
screen?
Shi, thank you so much for your exciting talk today again. And first question for you just came up. Can you see this?
Oh, I can't, sorry.
Okay, probably it's just coming up. It just takes a couple of seconds usually.
Oh, it's very small. I'm sorry.
The font size is too small to read.
Yes.
Okay. 크게 할 수 있나?
좀 크게 해주시고 그 사이에 한번 말씀해 주시는 게 나을 것 같습니다.
Okay. So I think I can read this for you just quickly. So the question is, in the phase 1B cohort planning, the first line DLBCL trial design includes the combination of R-CHOP with ROR1 ADC. Since R-CHOP has the cyclophosphamide and PBD, so they have the overlapping mechanism. So the concern here is that maybe there would be a toxicity and resistance. So can you comment on the component of the R-CHOP and PBD? Potentially they have the overlapping, the mode of action? Yeah.
Sure. Thank you. Thank you for your question. This is a very good question. Actually, as we designed the first line combination therapy strategy, this is one of the key factors for us to consider. So we choose to have CX-501 in combination without replacing any drug in R-CHOPs for the following reasons. As we know that oncowin, which is a microtubule inhibitor, contributes less critically to the R-CHOP's efficacy than cyclophosphamide and doxorubicin. It has, as we know, it has neurotoxicities, especially for the grade 2 or higher neurotoxicities, quite clinically challenging to manage. Actually, this risk is amplified with neurotoxic payloads such as MMAE, which is used in zero-V design. So this prompts Merck to replace Oncovin in their zero-V R-chip regimen, which apparently they remove Oncovin. And for cyclophosphamide, this remains an essential. We believe this is an essential drug for DLBCL's treatment efficacy, and its primary toxicity is unlike the Oncovin, which is neurotoxicity. The primary toxicity for cyclophosphamide is actually hematological toxicities, which we believe is more readily to be monitored and managed, such as we use the prior prophylactic growth factors, as well as those modifications than persistent neurotoxicity. So we believe that using a reduced dose of CX-501, which we will employ in our combination studies, the combination study, the dose of CX-501 will be lower than the monotherapy. We believe using this reduced dose, this can minimize the risk of overlapping hematological toxicities with cyclophosphamide. So by using this, we anticipate enhanced efficacy of CX-501 plus R-CHOP with manageable safety profiles supported by proactive safety monitoring and dose optimization.
Great. Thank you so much. 사실은 이게 약간의 테크니컬한 퀘스천이라서 조금은
This is a
쉽게
bit technical
풀어서
question,
말씀을
so let
드리겠습니다.
me easily explain the answer to you. In the BLBC hematological cancer, we use a cocktail including R-CHOP. So one of them is cyclophosphamide. You don't have to remember this. It's quite similar to the PBD in our ADC. It actually targets DNA. It also targets DNAs. So if two drugs target DNA, it might increase toxicity. There was a concern about that. So Dr. Shi explained another component of R-CHOP causes neurotoxicity. Merck uses MMA2 payload that actually causes neurotoxicity. So Merck could not use all of the R-CHOP. So from the cocktail, they had to remove it. But with our drug, we can use everything. Cyclophosphamide has a bit different MOA, but what's more important is this drug is not neurotoxicity. We can actually manage this toxicity because it is hematotoxicity. So our ADC shows very low hematotoxicity. So if you use a cocktail, toxicity might amplify, but our drug has very low hematotoxicity, and our ADC doesn't generate much hematotoxicity. So even if we add our ADC in the cocktail, it's not going to cause high toxicity. That was the rationale behind our explanation. Let's move on to the second question. Let's read the second question now.
Yes.
Okay.
Sure. So, thank you. Another good question. Yes, Merck's Zillow has already entered a phase three study. We are, although we are the global number 2 in the clinical development stage, considering the phases of the studies we are in, but we don't believe that we do have other advantages considering our profile. So for Zillow, as I mentioned in my presentation, it has around 30% of overall response rate in relapse and refractory diffusive large B-cell lymphoma as monotherapy. As we know, with this level of response rate, a single arm registration study is challenging globally. So for CS501, actually as monotherapy, it has demonstrated an overall response rate of around 50%, with a higher response rate in the tentative recommended phase 2 dose. With this response rate, we believe a single arm registration study is still possible in China as well as globally. So if currently we have initiated a phase 1B dose optimization study in this setting with a potential of a single arm registration. So if this pathway is feasible or workable, we believe we can register this drug with a much shorter timeline, comparing with a lengthy and costly global phase 3 study as initiated by Merck. So in addition, as we know that Merck's Zillow does not show efficacy activity in indolent lymphomas like follicular, like MZ, marginal zone, etc., and CLL. I think this is mainly because of the payload they use, which is MMAE, which is a tubulin inhibitor. As we know, tubulin inhibitor generally does not work in slow-growing malignancies like FL and CLL, etc. And for us, we do have shown some efficacy signal in the indolent lymphomas, which we believe put us in a development advantage position. So of course, we are actively exploring the follicular as well as CLL, etc., which we hope to see some more efficacy signals. And we also have some efficacy signals in solid tumors, which is, we believe CS501 is the first ROR1 ADC that has shown some signal in the solid tumor. This is a broader stage or broader indication than the lymphomas. And for Merck's phase 3 study, actually it's just initiated, and with over 1,000 patient plans and expected primary completion date of 2029, which is we believe is a long way to go. Currently, we are exploring the first-line combination with R-CHOP and the second, third-line combination with other standard of cares. I think with the if we identify or we detect some efficacy signal and prove the safety profile is favorable, we will initiate a phase 3 study very soon. We actually quite confident on CStone's development capability, especially to run the trials. So even for phase 3 studies, we believe we are not late. We still have time to catch up. Thank you.
Great answer. Thank you. 뭐 질문은 비교적 심플합니다. 그 머크
The
ROR1
question is very simple. Merck's ROR1 ADC is in phase 3 clinical trial, but this company joins later, but still it's going to be successful. Her answer was very clear. To summarize her answer, its efficacy is really great, and side effects will be lower, so it's very competitive, especially in DLBC, where ROR1 targets. When you use the combination therapies, this is my idea I'm adding to her answer. R-CHOP, when it comes to R-CHOP, you see 90%, 100%. You might think it's really amazing, but naive patients show about 80% response rate. So, but because of that, if you have a high ORR with the combination, it's not really amazing. But when it comes to refractory patients, you need to increase ORR. That's the key. So I'm giving this background to explain the key points of her answer. For refractory patients, when you use monotherapy, Merck's drugs ORR is only 30%, but our data shows 50%, sometimes even up to 70%. So to get approved, single arm monotherapy should be approved to FDA on top of combination therapy. So in their area already we are winning. Not just a hematologic cancer, there's a slow growing cancer like follicular lymphoma, CLL, marginal lymphoma. So for these slow growing hematological cancer, Merck's MME doesn't work really. Microtubule inhibitor works really well for fast growing cancer because they are targeting microtubules for cell growth. But our drug targets DNA, so it works really well for slow-growing cancers. So we can actually expand our indication to hematological cancers. Merck actually gave up on this area, but for the first time, we are using ROR1 in solid cancer, for example, lung cancer and pancreatic cancer. So this is the very first ROR1 cancer therapy that shows efficacy for solid tumors, because there was no precedence with PBD, because to be effective in solid tumors, toxicity should be low. At the same time, toxic level should be high. But because of the toxicity, they couldn't do that. But our drug has low toxicity. It can be really effective for solid cancer as well. Fourth point, it's speedy. Merck is in phase 3 clinical trial. It seems like they are ahead of us, but their completion is scheduled to be by 2029, so it's a long way to go. So with R-CHOP combination, we can register studies and we can enter phase 3. We can fastly catch up with them. So by 2000, before actually their completion, we can actually speedily catch up with them. That was the gist of our answer. This concludes the Q&A session. This concludes the morning session. The afternoon session starts at 1:20. 5 minutes before the start of the afternoon session, please be seated. Online live stream will resume at 1:20. This concludes the morning session. Thank you very much.